Welcome to this explainer. Okay, today we're opening the ultimate biological cold case file. We are traveling back in time, way back, over 700 million years to investigate our deep ancestry. Our mission? To figure out exactly how this spectacularly vast diversity of animal life, from microscopic flatworms all the way up to humans, actually arose from single-celled ancestors. And since we obviously don't have a time machine lying around, our primary forensic evidence won't just be rocks and fossils. We're going to dig deep, and I mean really deep, into the molecular biology of life. We'll be using highly conserved protein domains, microRNAs, and incredibly complex gene regulatory networks as our smoking guns. So here's our roadmap for this investigation. First, we'll map out the animal molecular blueprint. Then, we'll look at Chowan flagellates and cadarin proteins. After that, the Cambrian explosion clues, followed by blueprinting the body plan, and finally, we'll zoom out to the genomic tree of life.
All right, let's jump right into Section 1, the animal molecular blueprint.
Before we can really look at how animals diverge, we've got to understand the fundamental traits that unite this totally wild, diverse kingdom at the cellular and embryonic level. So, in most animals, reproduction produces a diploid zygote. This single cell immediately kicks off a process called cleavage. Think of it as a super rapid succession of mitotic cell divisions that literally skips the normal G1 and G2 growth phases of the cell cycle. And this whole rapid-fire process is initially driven by maternal messenger RNA that's already loaded right into the egg. This rapid division creates a multicellular embryonic stage called a blastula. It basically looks like a microscopic hollow ball. Then the real magic happens. A process called gastrulation folds one end of that embryo inward, kind of poking it inside out, filling the hollow space and producing the highly specialized layers of embryonic tissues that will eventually develop into adult body parts. This exact process is shared across nearly all animal taxa. It's our foundational molecular blueprint.
Now, you might be wondering, what actually tells those embryonic tissues what to become? Enter Hox genes. This is a super versatile family of regulatory genes containing what we call homeobox DNA sequences. You can think of Hox genes as a master evolutionary toolkit. These sequences code for homeodomains, specific protein structures that physically bind to the DNA. They act as powerful transcription factors, essentially turning other genes on and off like massive, complex molecular switches. Now, in the ancestors of complex animals, early homeobox genes were duplicated over and over again and repurposed over millions of years to yield a brilliant system for regulating development. From mapping out the anterior, posterior, or front-to-back axis of a tiny little fruit fly, all the way to the spinal column of a human being, these highly conserved molecular instructions are essentially running the exact same developmental software.
Moving on to Section 2, choanoflagellates and cadherin proteins. Let's shift our forensic investigation to one of the absolute biggest leaps in evolutionary history, the jump from single-celled organisms to complex multicellular animals. Now, this brings up a massive, really intriguing mystery. To build an animal, individual cells have to actually adhere to one another and communicate, right? But how did single, free-floating cells originally learn to stick together and talk to each other to form the very first animals?
Well, the answer seems to lie in our closest living single-celled relatives, the choanoflagellates. Researchers sequenced the genome of a specific unicellular choanoflagellate called Monocyga brevicalis just to look for clues. Basically, they were hunting for the origin of cadherin proteins. In animals, cadherins are absolutely crucial. They dictate exactly how animal cells physically attach to one another and participate in complex cell signaling pathways. And here's an amazing piece of molecular trivia for you. They're called cadherins because they are calcium-dependent adherents. That means they literally require calcium ions to function and glue cells together. Astonishingly, researchers actually found genes encoding for 78 protein domains in that single-celled choanoflagellate that we previously thought only existed in animals.
When we actually compare these proteins at the molecular level, we see something super fascinating. The ancestral choanoflagellate has a cadherin-like protein made of 7 distinct domains. And guess what? Animal cadherin proteins contain these exact same ancestral domains. But there is one absolutely crucial difference. Animals have an extra, highly conserved region incorporated right into their protein structure, the CCD domain. This specific domain is found only in animals. It is the unique molecular puzzle piece, the literal glue, if you will, distinguishing us from our single-celled ancestors. It proves that multicellularity evolved by basically rearranging existing protein domains and just adding a novel one. How cool is that? Okay, let's move to Section 3, the Cambrian explosion clues. So we have the toolkit, and we have the cellular glue. Tracing this chronologically through the fossil and biochemical record, we see a really fascinating timeline of how these blueprints exploded. Around 710 million years ago, we find chemical evidence of steroids that are uniquely produced by sponges, pointing to early animal life. Fast forward a bit to 560 million years ago, and we've got the Ediacaran biota, these soft-bodied multicellular organisms. But then between 535 and 525 million years ago, something incredible happens: the Cambrian explosion. In a total blink of geologic time, the oldest fossils of about half of all living animal phyla suddenly just appear. Hard, mineralized skeletons, complex predators, prey, the whole shebang. And by 365 million years ago, we literally have vertebrates walking on land. But what on earth triggered that massive burst of life in the Cambrian?
Well, there are a few major hypotheses. First, novel predator-prey relationships emerged, which likely drove a massive evolutionary arms race. Second, a significant increase in atmospheric oxygen would have allowed for much larger body sizes and higher metabolic rates. And third, and this is where it gets really interesting for us, drastic genetic changes affecting development occurred, specifically the origin of those Hox genes we mentioned earlier, and the addition of new microRNAs. Now, while environmental factors like oxygen and ecological factors like predators absolutely set the stage, it was really these deep molecular genetic changes that actually built the actors. And the hard data backing up this genetic hypothesis is just brilliant. Look at this. Sponges have an average of just 5.8 microRNAs and 25 cell types. Flatworms jump a bit to 35 microRNAs and 30 cell types. But vertebrates? We have a massive average of 147.5 microRNAs, which corresponds to over 172 distinct cell types. Now, these microRNAs work by physically binding to target messenger RNAs to silence them, acting as incredibly precise fine-tuners of gene expression. So when you look at the numbers, there is a deeply compelling positive molecular correlation between the diversity of microRNAs an animal has and its resulting morphological complexity.
Ultimately, as researchers note here, genetic changes affecting development, such as the origin of Hox genes and the addition of new microRNAs, facilitated the evolution of new body forms. So the big takeaway here? The hidden engine driving the incredible diversity we see in the fossil record wasn't just bigger teeth or more oxygen in the air. It was deep molecular gene regulation. That was the real game changer.
Next up, Section 4, Blueprinting the Body Plan. Now let's examine how these genetic changes actually manifest into physical, three-dimensional bodies, or body plans. A fundamental feature of animal bodies is their symmetry. Radial symmetry is kind of like a flower pot. There's a top and a bottom, but no left, right, front, or back. This perfectly suits a sessile or drifting lifestyle. You know, like a sea anemone just meeting its environment from all sides. On the flip side, bilateral symmetry has a two-sided orientation. You have a distinct front, back, top, and bottom. This evolutionary leap is huge because it equips animals for active, coordinated movement. Plus, it allows for cephalization, which is just a fancy word for the heavy concentration of nervous tissue and sensory equipment right at the anterior or front end of the body.
Going a layer deeper, literally, let's look at tissue organization. Bilaterally symmetrical animals are called triploblasts, meaning their embryonic cells form three distinct germ layers. Think of it exactly like an intricately constructed sandwich. You have the ectoderm securely on the outside, forming the outer covering and nervous system. You've got the endoderm completely on the inside, lining the digestive tract. And firmly sandwiched right in between is the mesoderm. This forms the muscles and most internal organs, as well as true body cavities, or columns, that cushion and protect our organs while allowing them to grow completely independently of the outer body wall.
Let's rewind for a second back to those very early embryonic cleavage stages we discussed earlier. Triploblastic animals generally fall into two different camps based on their molecular development. First, you have protostome development, which features spiral, determinate cleavage. This basically means the developmental fate of each embryonic cell is rigidly cast incredibly early on, often driven by the unequal partitioning of molecular determinants, like specific mRNAs and proteins, in the cytoplasm. For example, if you separate the cells of a snail embryo at the 4-cell stage, they'll just fail to develop. But deuterostome development, which includes us vertebrates, features radial, indeterminate cleavage.
And let me tell you, this indeterminate cleavage is an absolutely phenomenal molecular trait. It means that if early embryonic cells are separated, each individual cell retains the full genetic capacity to develop into a complete, healthy embryo. This exact cellular feature is literally what makes human identical twins possible. Our early cells aren't just rigidly locked into their fates right out of the gate. Finally, Section 5, the genomic tree of life.
Having analyzed the cellular glue, the complex gene regulation, and the developmental body plans, it is time to zoom all the way out. Let's look at the entire phylogenetic tree through our modern genomic lens. When we look at bilaterally symmetrical animals, genomic data groups them into three major clades. First, Deuterostomia, which includes hemichordates, echinoderms, and us, the chordates and vertebrates. Second, Lophotrochozoa, which are entirely invertebrates like mollusks and annelids, named for their ciliated tentacles or unique larval stages. And third, Ecdysozoa, which are invertebrates like arthropods and nematodes that actually secrete and shed tough external exoskeletons in a process called ecdysis.
Now, for a long time, biological classification relied heavily on physical body plans. But today, our absolute ultimate forensic tool is whole genome sequencing. By analyzing complete genomes, looking at highly conserved sequences, and utilizing molecular clocks, which basically track the mutation rates of specific genes over time, scientists are able to trace lineages back hundreds of millions of years. We're constantly refining and reshaping our understanding of evolutionary relationships. It just goes to show that science is a beautifully dynamic process of continuous inquiry.
And because of these incredible molecular tools, there are massive, super exciting debates happening right now. Traditionally, simple, tissueless sponges were considered the undisputed basal metazoans, basically the very first animals to diverge from the evolutionary tree. However, recent phylogenomic data, which compares massive data sets containing thousands upon thousands of genes, is totally flipping this traditional script. These deep, in-depth analyses suggest that cletophores, or comb jellies, which actually do have tissues, might be the true basal metazoans. It's literally an intense battle for the base of the animal tree, and it's still unfolding as we sequence more and more data.
Which leaves us with a thrilling mystery. As our technology improves and we peer deeper and deeper into the molecular codes of life, if our genomic tree is still changing today, what molecular clue will rewrite our history next? The evidence is hidden in the DNA all around us, just waiting to be discovered. Thanks for joining me on this explainer. Keep your curiosity alive, and I'll see you next time.